Hello champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about sexual reproduction in flowering plants. And the topic that I chose is Microsporangium. Well, a clear header over here. Basically, we would be not giving too much lectures for this particular topic. Why? Or this particular chapter, not topic. Why? Because it gave you 2 questions the previous year. Okay, so 2 questions into basically 4 marks is what? 8 marks. Okay, 8 marks. So obviously you can think in 2 ways. One is that I won't study this chapter, I'm okay to lose my 8 marks. Second one is that I can invest just a little bit of amount of time for this chapter and I can gain 8 marks or more, because nowhere it is written that it will obviously always give just 2 questions. So it can be more as well. So why waste your time by thinking that you should not be studying this chapter? You should be definitely not missing this chapter, because 8 marks means a lot if you are preparing for a competitive exam. Now in the previous session we covered human health and diseases, which gave 16 marks, and we also covered human reproduction with 20 marks and reproductive health, lots and lots of things. So if you want the PDF of all these things that we have studied, you can join our WhatsApp channel. The link is given in the description box. Yeah. Now over here children, the first question that I want to ask you is, do you know the flower really well? So we know we have studied the structure of the flower for like years. From 6th class onwards we might be studying, but we should be knowing the structure. What is the colored part over here? What do you call it? Petal. Now, petal is that part of the plant which is responsible for attracting the pollinators. This question has come. What is that part of the plant which is responsible for attracting pollinators? Petal, sepal, only petal, petal and sepal. So things like that. So you should be knowing is just petal. Over here, the green part that you see, they are known as what? Sepals. They are the ones that protects the flower in the bud stage. Okay. Now going ahead, you have a long stalk-like structure which is there. What do you call it? Pedicel, children. And pedicel, if you see, it has a swollen structure at the top. What do you call it? You call it thalamus. You call it thalamus, children. Okay, now going ahead, what do we have? We have, and thalamus is important. I'll tell you why later on. So this particular structure which is there, which is a lobed thing at the top and a very long stalk-like structure, you call it stamen, and this guy is our today's hero. We have to talk about stamen today. Okay, going ahead, you have another structure, which is the female reproductive structure. What do you call it? Pistil or carpel. Now inside pistil or carpel, what do you have, children? You have ovule, which will be having the female gamete, right? Now why did I say that thalamus is important? Because when you study female reproductive system, there you will understand that there are certain plants in which the pistil is not attached to the thalamus, rather than it is attached to the petals, and that is a whole different condition. Okay. Now petals, when they come together, what do you call this, children? Petals, a group of petals. What do you call this? Corolla. A group of sepals, what do you call it? Calyx. So these are just little information that you should be definitely knowing if you are going to start off with sexual reproduction in flowering plants. Now let's get ahead with our topic, and I want to tell you, I'll be making the diagrams, explaining you things, and then we will be reading the NCERT. Okay. So over here, the structure of the stamen, it looks like this. So we know there is a big lobed structure, and then there is a very long structure, stalk-like structure, which is going to land on the thalamus. Yeah, this long structure that you see, let's start with it. It's known as filament, children. It is what? Filament. And filament, as you see, this area of the filament, it is known as proximal region. Proximal region. Proximal region as in the one that is in proximity with the flower base. That is the thalamus. This region, it is what? Distal region. Basically, the one that is far from the bottom of the flower. Okay. Now, at the distal region, what do you have, children? What is this particular structure that you see over here? It is the anther. It is what? The anther. And as you can see, anther is what, children? It is first feature of anther. It is bilobed. There are 2 different lobes. This is one lobe and this is the second lobe that you have over here. Now, I am saying, apart from being bilobed, the structure is almost like this. They also have a middle cut in between. Okay, they have something like this, some grooves like this in between, and if you cut the anther and you look for this from the top, it would appear something like this. So basically, Okay. Yeah, it have a structure like this, where this is, children, this is one lobe and this is the second lobe, which is why I say it is what? Bilobed. This is one lobe, this is second lobe, and each lobe is also divided into two regions. What do you call this? You call this as theca. You call this as theca. One is theca, this is another theca, this is other one, this is the other one. So each lobe, if I say, if I talk about one lobe, can I say it is dithecous? It has two thecas, right? So each lobe, basically children, it is what? Dithecous. Now inside each lobe, you will be finding some cells like this over here, over here, over here, and over here. What do you call it? You call it Microsporangium. You call it Microsporangium, which is the one responsible for the formation of what, children? Pollen grains. So they would be the one that would be forming the pollen grains. But they are not alone. They are surrounded by layers of cells. There are different layers of cells. So let's see. The first layer, which is over here. Okay. What do you call this layer as? Epidermis. You call this layer as epidermis. Now going ahead, you have next layer. What do you call this layer as? Endothecium layer. You call this as endothecium. Now, there is another layer. This layer is known as, is it visible? Yes, middle layer. So you have three layers: epidermis, endothecium, and a middle layer. So middle layer has to be in middle of something, right? So middle layer is in between, one layer like this, okay, which you call as tapetum. What do you call it? Tapetum. Okay. Now tapetum, there is a very important feature about tapetum. They do not have one nucleus, they have two nucleus. So they are binucleate. They are binucleate. So here they have underwent nuclear division, but the cell division has not been taken place. Cytoplasm is not divided, just the nucleus divided. So that is why they have what? Binucleate condition over here. Now, what is the use of tapetum and how it is different from all these layers over here? Basically, tapetum is the one that is responsible for nourishment of the microsporangium. It is responsible for the nourishment of what? The microsporangium. So these both guys, they are a team. We can say they are a team. But then what is the use of all these three layers which are over here? Epidermis, endothecium, middle layer. They are there for protection. Agreed? They are there for protection for obvious reasons. Apart from protection, no children, they are there for dehiscence of anther. What do you mean by dehiscence of anther? Have you heard about dehiscence before? Dehiscence basically means there is breakdown of anther, and if the anther is getting broken down, basically if it opens up, what will be released, children? The pollen grains which are present over here, they will be released outside. That is what you mean by dehiscence of anther. So you have epidermis, endothecium, middle layer, and tapetum. These are the four layers that you have to remember for sure, and they ask you also. They can give you the picture, they can ask you for what's labeling things. Okay. Now how can you remember it? You know there is ENT doctor, yeah, ear, nose, and throat, right? Similarly, you can remember EMT. EMT. I know it does not make much sense, but still somehow it might help you. So E for epidermis and endothecium. Okay, epidermis will be always outside, epidermis. Okay, so it will be always outside. So epidermis is the first one, then endothecium, then middle layer, and then our tapetum. The tapetum is the one that provides nourishment, children. Remember that. Yeah. Now that is done. Agreed? Now microsporangium, if you see, microsporangium, no children, it is made up of so many cells. Okay, and what do you call them? You call it sporogenous tissue. Sporogenous tissue. So microsporangium is a place where the pollen pollen grains would be formed. It's not the pollen grain, it's a place where the pollen grains will be formed. And microsporangium has lots and lots of what? Sporogenous tissue. Okay. Now, what happens to the sporogenous tissue? Let's see that. Basically, this is the structure that you see over here. I have made epidermis, endothecium, the middle layer, and then comes what say microspore mother cells, which is there inside. Where does the microspore mother cell come? We'll come to that as of now. We'll come to that layer. Okay. Now, tapetum is the innermost layer, and inside that you see there is sporogenous tissue over here, which is the innermost layer. Right. Now, let's see. We'll come to this also. Okay. Now, over here in the sporogenous tissue, in the sporogenous tissue, there are cells which you call as what? Pollen mother cell, which is PMC, or you also call that microspore, miscoro. Oh, tell me. Teacher has gone nuts. So, over here, yeah. Why? Microspore mother cell or MMC. Okay. Now, these are cells. They are going to form, as you can see, mother cell. Basically, they are going to form the babies over here. And who are the babies' children? They would be our pollen grains. Do you think it's pollen grains? No. There's a suspense over here. So, basically, the pollen mother cell or the microspore mother cell, it undergoes meiosis. It undergoes what, children? Meiosis. And, sorry. Just a second. Okay. So, the pollen mother cell or the microspore mother cell is going to undergo meiosis, meiosis, yeah, and meiosis, you know, it is divided into two stages, meiosis 1 and meiosis 2. So, it undergoes meiosis 1 and then it forms 2 microspore. This microspore will again undergo meiosis, that is meiosis 2. And meiosis 2, what will happen? This cell will divide into 2, this cell will also divide into 2. So, basically, what do we have, children? We have 4 microspore over here. So, we have 4 microspore, and this microspore, 4 of them, what do you call it? Microspore tetrad. Okay, what do you call it? Microspore tetrad. Now, this microspore will be the one that will be responsible for the formation of pollens. I know you want to kill me, but it works like that only. Okay, so sporogenous tissue is there, children. The sporogenous tissue is made up of pollen mother cell or microspore mother cell. Yeah, the microspore mother cell or the PMC basically undergoes meiosis. There are 2 rounds of meiosis, meiosis 1 and meiosis 2. At the end of meiosis 1, you get 2 microspores. 2 microspores. At the end of meiosis 2, this one also divides into 2. You get microspore tetrad. Now, this is there are 4 microspore. Agree? Now, each of them can give rise to a pollen. Okay, each of them can give rise to a pollen. So, over here, microspore can now give rise to a pollen. Each of them basically are going to act like a pollen. So, this is something that we need to read first, and then probably we could understand things better. So, over here, I have, formation of pollen grain. You read this. As each of the sporogenous tissue is capable of giving rise to a microspore tetrad, as you see, okay, each one is potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is known as microsporogenesis. So, what you saw now was microsporogenesis. Okay, the microspores, as they are formed, are arranged in a cluster of 4 cells, the microspore tetrad. We saw that was the end stage, right? As the anthers mature and dehydrate, the microspores dissociate from each other and develop into a pollen grains. Develop into what? Pollen grains. So, these structure that you see over here, these are just going to form what? Into pollen grains. So, basically from microspore tetrad, how many of them can you receive? 4 pollen grains. 4 pollen grains. Okay. Now, still, do you have the male gamete? Still, do you have the male gamete? No. Still, you do not have the male gamete. Now, if we see the structure of the pollen grain, if you see the structure of the pollen grain, it looks like this. Basically, the pollen grain is like this. Okay, this is just one cell. This one cell will undergo mitosis, children. It will undergo mitosis. Okay, so basically when undergoing mitosis, a cell has to divide into two other cells. Agree? But over here there is a little twist. Over here there is a little twist. Instead of dividing into two separate cells like this, okay, instead of dividing into two separate cells like this, there is something else happening. So when it undergoes mitosis, there will be a cell like this, and inside the cell only there will be another cell. So this cell does not move out, this cell is just there. So here you will be having a nucleus for this cell, here is the other cell that you have. Okay, so basically the pollen which was there, children, the pollen which was there, it underwent some process which is known as what? Mitosis. Yeah, and now what is happening? You have two cells. This particular cell that you see over here, you call it vegetative cell, and this cell that you see over here, this is the generative cell. Why generative? Why vegetative? Because generative cells are the one that is going to form the male gamete. Okay, they are going to generate the male gamete. Still you do not have the male gamete, you are going to form the male gamete. So when next mitosis happens, so this is first mitosis that happened. Now there will be one more mitosis that happens, okay, where you get two cells, and these two will be what, children? Both of them together they will be known as what? Male gametes. So you have two male gametes would be formed from the one pollen. So you have two male gametes formed from one particular pollen. If you didn't understand the whole thing, I can explain to you. So basically, if you see, you had sporogenous tissue, children. Sporogenous tissue. Agree? Each cells of the sporogenous tissue are PMCs, pollen mother cell, or basically microspore mother cell, right? Now, these microspore mother cell will think that, okay, we have to do what? Meiosis. So, meiosis 1 happens, you have two microspores. Two microspores. Still, okay, I do not know if we can say microspores. So, you have two cells. Now, these cells again, they will undergo meiosis 2 because meiosis has two phases. So, it is meiosis 2. Now, over here, they will be forming, this cell will divide into two. This cell will also divide into two. Now, you have 1, 2, 3, 4 microspores, and these 4 microspores, what do you call it? Microspore tetrad. Since there are 4 of them, you call it microspore tetrad. Now, these microspore, okay, each microspore which is there over here, this is the one which is going to act like a pollen, So, each one of microspore, they are going to act like one pollen. Now, this pollen, if you see, it also has to undergo mitosis. When it undergoes mitosis, you have one cell, vegetative cell. Inside the cytoplasm of the vegetative cell, you have another cell which is generative cell. Now, that's not all. When you have to form the male gamete, this generative cell again undergoes a division which is known as what? Mitosis. It again undergoes mitosis. This mitosis is mitosis 2, right? And now when it undergoes mitosis, you get two male gametes. Okay, is this clear? So, for formation of the male gametes, what do you need? You need one whole cycle of meiosis. You need one meiosis and two mitosis. Please remember, it's a very, very, very important question. Okay, is this clear? Now, over here we can explain the structure of the pollen. Okay, so I am going to erase this. I am going to erase this particular thing, and I am going to explain you the structure of the pollen. So, instead of this, I'll write structure of pollen. Okay, see very carefully, okay. So you have, as I said, two cells. One cell is floating in the cytoplasm of the other cell. This is the generative cell and this is the vegetative cell. Agree? It has its own, this is a cell, so obviously the first layer that we will be having will be the plasma membrane. Will be what? The plasma membrane. The plasma membrane now has two layers outside. So pollen, no, you know, pollen basically they are resistant to a lot of things. It stays in our nature for so long, and after some time probably it might get damaged, but mostly it does not. So basically what happens, it does not get damaged because it has two layers, and I don't want to make it in green again. Let's use white. So the inner layer that you see over here, it is known as, children, it is known as intine, and intine is made up of cellulose and also pectin. It is made up of cellulose and pectin. This is the inner layer. Now outside this particular layer that you see over here, you have some layer like this. What do you call this layer as? Exine, because it is outside, exit, okay, you can remember like that. And this is made up of something very unique which you call as sporopollenin. What do you call it? Sporopollenin. This, children, know it is highly resistant to any kind of damage. So no damage can happen to the pollen grain if it is made up of what? This sporopollenin. So exine layer, it is always made up of something that is known as what? Sporopollenin. Agree? Now if you see, there are some pores that I made over here. What are these pores, children? These pores that you see, they are known as germ pores. They are known as germ pores. So why are germ pores there? Basically germ pores are there in the exine because you want the pollen tube. So pollen, when it goes and lands on the stigma of a flower, when it goes and lands of the stigma, basically in the female reproductive tract, right? When it goes over there, not female reproductive tract, in the female reproductive part of the plant, when it goes there, this pollen forms a pollen tube and it sends the male gamete over here. So to send the male gamete over here, to make the pollen tube, you need a pore in this, and that pore is what? Germ pore. So imagine with the help of this pore, no, what will happen? This particular cell, the green cell that you see over here, the vegetative one, okay, the vegetative one will slowly form the pollen tube. If I rub this, everything will go, that's why I'm not rubbing. And then, as I said, this cell is going to undergo mitosis. You have two cells, one cell will go and then the next cell will go. So the gametes would be travelling down with the help of the germ pores. The pollen tube will be formed over here itself and the gametes could easily move out. Who helps in the formation of the pollen tube? The vegetative cell. The vegetative cell help in the formation of the pollen tube, and the generative cell, they divide, they undergo mitosis, they become two male gametes and the two male gametes go. Now, when does this division of male gamete happen? So in most of the, in most of the plant, 60% of the angiosperms. 60% of the angiosperms, as in the flowering plants, okay, they have the pollen in this particular state. They have the pollen in this particular state where the division has not taken place yet. Where the division has not taken place yet, there is just one generative cell which would later divide into two, okay. When it is released, when the pollens are released, it is in this particular state. Now when it goes and lands, the division and everything that happens there. Now, 40%, rest 40% of the angiosperm, in them division already took place, okay. Second, mitosis already took place and you will be having what? Two gametes. So they will be, now you only think, okay, if this is the case, then it is what? Two-celled stage. This particular case is what? Two-celled stage. There is only one generative and one vegetative cell. Now over here, if it is dividing into two, if this particular thing is dividing into two, then there is 1, 2 and 3. So can I say when it forms the two gametes after second mitosis, in 40% of the angiosperm, it is released in 3-celled stage, because you have to count that this is divided and now you have 2 of them. So 1, 2, 3, 3-cell stage. Will you be able to remember it? It's a very easy thing. It's not a very humongous big thing. It's a very easy thing. Now children, always remember that pollen, no, one particular pollen which has the capacity to give rise to male gamete. Because of that particular reason, I call that pollen is what, children? It is the male gamete. Pollen is what, children? It is the male, it's not male gamete, male gametophyte. It is the male gametophyte. So if you are asked what is a male gametophyte, it is the pollen. Okay, if you are asked what is a male gamete, it is basically the male gametes that are being produced inside the pollen. Okay, so since it is produced inside the pollen, you call this structure as male gametophyte. Agree? Now one more very important thing. Now when I say meiosis and everything has happened, obviously you need to know the ploidy of the cell, how many number of chromosomes are there. So over here, the sporogenous tissue is made up of cells, and all the cells basically, children, they are diploid. They have diploid, that means double the number of chromosomes. Okay, now that is the usual number of chromosomes. Now when these cells, they undergo meiosis, now when they reach first meiosis stage, they become what? Haploid. They become what, children? Haploid. There will be half the number of chromosomes. Why half is happening? And okay, going ahead, in meiosis 2 what will happen? Does it get more half? No. In meiosis 2 the number of chromosomes remains the same. So if it was haploid over here, in meiosis 2 also it will be haploid. That means microspore tetrad, each of them will be having what? Haploid nucleus. Over here that means what? The pollen will also be having what? Haploid nucleus. So this will be a haploid cell, this will also be a haploid cell. Why is it a haploid cell, children? Why is it N, not 2N? N means haploid, half the number of chromosomes, right? And why is it not 2N? Because you only imagine, the pollen grain will be having the male gamete. Male gamete has to be haploid. The female gamete also, if it comes to be haploid, both of them they combine together to form a diploid organism. We want diploidy. Now you only think diploid is coming, and then comes together, more number of chromosomes will happen, right? For example, imagine, just imagine. Okay, you have a 22 and 22 over here. Together they come, it becomes 44. Agree? Now the whole plant body is having cells of 44 number of chromosomes. But the male gametes, if they have the number of chromosome, for example, they are giving, instead of 22, they are halving it and giving just 11. Over here also they are giving 11. Now 11 plus 11 becomes what? 22. You didn't get it? Because I know I made it very confusing. So basically not 44. Basically, if you think as a human being, we have 46 number of chromosome, okay, in a normal cell. Now each cell will be having 23 chromosome pairs, okay, which is why we have 46. Now if you just imagine, from both the parents we got 46, the total will be what? From both the parents, if both the parents gave us 46, the total will be 92. Agree? So our cell will be, zygote will be having 92 number of chromosomes. Is it okay? No, because you double the amount, it's double the amount of DNA. We do not want that. We just want 46. So for making it 46, what do we have? We have to make the gametes haploid. So there will be 23 combines together 46. Get it? Similarly, the same thing happens with plants also. The plants, basically they are diploid over here. They will have their certain number of chromosomes. It will give half, and now this half and half, when it combines together during fertilization, during fusion, it becomes full, it becomes diploid again. So just always be ready to imagine that this is diploid, and rest everything, once it starts meiosis, it will be obviously haploid. Okay, will you be able to remember it? Now let's solve the questions that are present over here. Okay, first question. Proximal end of the filament of stamen is attached to what? Proximal end of the filament. They are asking you, this is the anther, okay, this is the anther with bilobe, and this is the filament. Where is the proximal? This was the proximal end. Where was it attached to? I told you it is attached to the thalamus or the petal. This is why I gave this information in the starting of the chapter, that thalamus can also be taken place, or petal. Both of the places the filament can come and attach. Okay, this came in NEET 2016. Going ahead, we have next, male gametophyte in angiosperms produced. Male gametophyte, that means you are talking about what? Tell me, what is a gametophyte? You are talking about pollen grains. So over here, the male gametophyte is going to produce what? Tell me. Single sperm and 2 vegetative cells, or 3 sperms, or 2 sperms and a vegetative cell, or a single sperm and a vegetative cell? You tell me. Is it producing 2 vegetative cells by any chance? No, it is just 1 vegetative cell, and then you have 1 generative cell which again divides into 2 male gametes. So 2 male gametes as in 2 sperms and 1 vegetative cell. So this is the correct answer. This came in the year 2015. See, this is how question is framed. Which one of the following statements is correct? Endothecium produces the microspore. Endothecium was the one that covers. It is not producing the microspore. Tapetum nourishes the developing pollen. Hard outer layer of the pollen is known as intine. I think it's known as exine. Right? Sporogenous tissue is haploid. Can it be haploid? No, it is what? Diploid. I told you sporogenous tissue are made up of PMC. All of them, they are diploid. They undergo meiosis and now it becomes haploid. Right? So basically this is wrong. This is what? They are diploid. This is exine, and this is also wrong because endothecium does not produces microspore. It is just the outer layer. Tapetum nourishes the developing pollen. That is correct. This came in the year 2013. So if you are not aware about this point, how can you get the answer? So you should be knowing all this thing. Going ahead. Okay, now this we have to study. So let's study. So the picture, I'm thinking that this thing probably it's not of any use if we read it as of now because we have already completed, but you can give a quick read about it. Let's see what we need to read. All this thing we have studied. Okay, let's see, read this because this is important. Pollen grains are generally spherical, measuring 25 to 50 micrometers. Please remember this thing. 25 to 50 micrometers. Okay, please remember it's a diameter. It has a prominent 2-layered wall. The outer layer called the exine is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollen is so far known. So that is the most important thing. Pollen grains exine has prominent apertures as in hole. What do you call this? Germ pores, where sporopollen is absent. So here sporopollen won't be there, which is why there will be pore. Okay, pollen grains are well preserved as fossils because of the presence of sporopollen. You can preserve it as a fossil because it can stay for so long because of sporopollen. It is that much protection that it is offering. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of the germ pore? I told you what is the function of the germ pore for the formation of the pollen tube, right? Now, the inner wall of the pollen grains is known as intine. It is a thin and continuous layer made up of what? Cellulose and pectin. Pectocellulose could also come. Okay, so understand, use your brain there. So pectin and cellulose. Now, the cytoplasm of pollen grains is surrounded by a plasma membrane. I told you, obviously if it's a cell, it has to be provided by plasma membrane. Then comes in the next sign. When the pollen grain is mature, it contains 2 cells, the vegetative cell and the generative cell. Okay, the vegetative cell is bigger and has abundant food reserve and large irregularly shaped nucleus. So this particular cell that you see over here, it will be having irregularly shaped nucleus and also good amount of food reserve because you want to nourish the male gametes. Now, the generative cell is small and floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this 2-celled stage. I told you, only generative cell and vegetative cell. In the remaining species, that is 40%, the generative cells divide mitotically to give rise to 2 male gametes before the pollen grains are shed. And this is 3-cell stage because you will be having 1 generative cell and 2 male gametes. So 3 cells in total. Agree? Now this is a structure. If you want, you can just have a quick look, and over here, yeah, that's all. And going ahead, yeah. Now pollen grains and allergy. This is some extended topic that we need to know. Let's see. Pollen grains of many species cause severe allergies and bronchial affliction. Bronchi, you know the part of the lungs, right? So if this is the lungs and this is the trachea basically opens into bronchi. Bronchi later divides into bronchioles. Bronchioles later divides into what? Your alveolus. So it can lead to bronchial afflictions as well. Now, it can also lead to chronic respiratory disorders like asthma, bronchitis, etc. It may be mentioned that parthenium or what do you call it, carrot grass, please remember it. Parthenium or carrot grass that came into India as contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. So this is one very important thing. Parthenium, please remember the name. Carrot grass, they are the name for this. It has caused a huge amount of ruckus by causing pollen allergy. Going ahead, pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets also. So pollen grains, obviously, you know, there is generative cell which is going to give, vegetative cell that is going to give nourishment and everything. And obviously, since it is giving nourishment, it has to be rich in something, right? So that is why this particular pollen tablets are given as food supplement also in Western countries. A large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has to be claimed to increase the performance of the athletes and the race horses as well. So it is very much useful for the sports people, for us, for everyone, because it has claimed to be a good source of what? Nutrients. Okay? Going ahead. So please remember parthenium over here. Parthenium is also known as carrot grass. And pollen viability and storage. What do you mean by viability? The ability to fertilize. The ability to fertilize. Over here they have given certain examples of the plant species. You have to remember it. When once they are shed, that is pollen grains, they have to land on the stigma before they lose their viability, as in the ability to fertilize, if they have to bring about fertilization. How long do you think the pollen grains retain for viability? It is different for different plants. The period for which the pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and the humidity. In some cereals from here, such as rice and wheat, pollen grains lose viability within 30 minutes. So you can just imagine rice and wheat, both of them, they are crying because they lose their viability within 30 minutes. So before that, fertilization could happen, should happen, not could happen, should happen, okay. Now, in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. This is again important, RLS. Okay, so over here, right, left side, you can remember like that. Rosaceae, Leguminosae, I'm not writing everything because it's there, and Solanaceae. All of them, basically, they can remain viable for months. It is again a very important piece of information. You may have heard that they maintain viability for, okay, you may have heard of storing semen, sperms of animals, including humans, for artificial insemination. Artificial insemination, what do you mean by that? Basically, for human beings not able to inseminate naturally, the semen or the sperm is not able to reach the female genital tract. In this particular point of time, you use artificial insemination, where either this particular semen is collected and been put into the female genital tract with the help of certain techniques, okay, or else the semen is put inside the egg, and then this whole thing is planted into the human body. So this is what you mean by artificial insemination, and you might have studied this in reproductive health. Yeah, it is possible to store pollen grains of a large number of species for years in liquid nitrogen. Degree is very much important, okay. It is not 1960 degree, it is not it is 196 degrees Celsius. It is what? 196 degrees Celsius, okay, please. Now, in liquid nitrogen, because only liquid nitrogen, in that only it can be stored. Now, such stored pollens can be used, can be put in pollen banks and then used in crop breeding programs. So, for example, just imagine that this particular plant is going to get extinct. Now, if we have the pollen of everything and after years we try to plant it, obviously we can get a good yield and that won't be extinct anymore. So, basically for crop breeding programs and everything, we can use this particular pollen. I hope this is completely done, and now we can do the question that was there already. Where is the question? Yeah. Which one of the following statement is not true? Pollen grains of many species cause severe allergy. It causes. And the one that we know is parthenium and carrot grass. Stored pollen in liquid nitrogen can be used in crop breeding programs. Yes, we can. What is the degree? 196 degrees Celsius. Tapetum helps in the dehiscence of anther. Exine of pollen grains is made up of sporopollen. It is correct. Tapetum, does it help in dehiscence of anther? No, it provides nourishment. So this is wrong. Okay. Now, pollen grains can be stored for several years in liquid nitrogen, having a temperature of what, children? 120, 80, 190. If I would have come, then what would have you done? It's 196 minus 196. Okay, not 196, minus 196 degrees Celsius. And with this, we have completed the microsporangium. I hope that this was helpful for you. Let's put the tick mark over here as well, so that we remember it later on. Which one of the following statement is correct, children? Tell me. Tapeworm nourishes the developing pollen. This is correct. Pollen grains can be stored for several years in liquid nitrogen. Where? Minus 196 degrees Celsius. Which one of the following statement is not true? You know, tapeworm helps in dehiscence of anther. This is not true, because tapeworm helps in nourishment. We just studied it. Proximal end of the filament of the stamen is attached to what? Proximal, that is lower end. Okay, it will be attached to thalamus or petal. Male gametophyte in angiosperms produces what, children? Gametophyte, that is the pollen. It will produce 2 sperms and 1 vegetative cell. Done. So that's all about microsporangium. All this type of questions have come. Apart from that, they have also asked about the ploidy. Okay, what is the ploidy of sporogenous tissue? It is diploid. What is the ploidy of male gametophyte? It is haploid. Okay, so you have to think for that. Now going ahead. In angiosperms, all the 4 microspores of tetrad are covered by a layer. Not male gametophyte, I was talking about male gamete. Okay. In angiosperm, all the 4 microspores of tetrad are covered by a layer which is formed by. You have to tell me. I am not going to help you. Okay. So tell me in the comment section, and in the next session I will be explaining this to you as well. So that's all, children. I hope that this particular video was useful for you, and if it was useful, do not forget to subscribe to the channel as well. See you on the next day, where we will be talking about Microsporangium. Yeah, bye bye, take care.